Hi, everybody. Okay, so at this point, we have talked about how DNA makes mRNA transcription in the nucleus, and that mRNA leaves, goes to the ribosome, and determines the order of tRNAs coming in to build a particular protein with a particular sequence of amino acids that makes you look the way you do. We've also learned that if the DNA is changed, a mutation, the mRNA will be changed, and that will change the sequence of amino acids and therefore the trait in many cases. Today, we're going to talk about how scientists have taken advantage of this knowledge to manipulate the genes in organisms to have traits that they wouldn't otherwise. That's called genetic engineering. We will later also talk about cloning and artificial selection in this note packet. All right, but today we're going to talk about genetic engineering. So we can take a particular sequence of DNA and insert it from one organism to another. So in other words, let's say we have a cow. We could cut out the DNA of the cow for its insulin and replace it with a gene from a human with insulin. So we could cut out the cow's DNA and put in the human DNA. And if we do that, if we take out the cow's DNA, it will no longer make cow mRNA and it will no longer make cow insulin. If we take out that DNA and we put in human DNA, then that human DNA will make human mRNA inside the cow. And the human mRNA will determine the order of amino acids and make human insulin. We don't do that with cows so much. We do it with bacteria, though. And that's genetic engineering. So anytime that a scientist takes a gene out of one species and puts it in another, we have genetic engineering. Sometimes this is done for rather silly reasons and sometimes really useful reasons, but no matter when it's done, it's pretty controversial at times. So for example, a silly reason for it is this fish right here. This fish, you can see it at many pet stores. You can find the natural zebrafish, and obviously it's called a zebrafish because it's got stripes. So some folks thought, oh, we could really appeal to consumers and sell more fish if we could make them glow in the dark. How cool would that be? So they took the genes from something that glows, which is coral. They took the genes out of coral and cut it out of the coral, an animal that lives in the ocean that can be very, very vibrant colors, and took that gene and put it into the zebrafish zygote. And so they only had one cell. They could take the DNA out of the zygote or put in the gene for bright color proteins from the coral into the zebrafish. And now you have things that you find in the pet stores today you could find called neons. They're actually zebrafish, but they have the genes from the coral that allows them to make this glow in the dark, outrageous color. That's genetic engineering or gene modification. And there's some questions about whether that's really a responsible use of such powerful science. But nonetheless, we do it. So to kind of go through how that works, we've got genetic engineering is when the DNA is transferred from one species or one organism to another. And if you change the DNA, then you're going to change the mRNA, and then you're going to change the protein. When you do change the DNA, the new combination of DNA from two different species is called recombinant DNA. That's a fancy way to say the DNA has been recombined in a new way. You have some DNA from one organism and some DNA from the other. In this case, you have some from the coral and some from the zebrafish together. And once it's together, it's called recombinant DNA. Now that this zebrafish zygote has genes from the coral, with this new DNA, it can make a new protein from the coral. Now, how that's all done, there's special enzymes. Remember, enzymes control chemical reactions. There are special enzymes that will cut DNA in specific spots and other enzymes that will paste it together with others to make recombinant DNA. And so it's enzymes that will control these reactions. And scientists have identified various restriction enzymes, particular types of enzymes that will make these cuts and these pastes to allow them to cut DNA from one thing to another. So let's look at this in a picture form. So if you have here a human cell and you have here a bacterial cell, you could take a gene from the human, cut it out with enzymes. You could take the DNA from a bacterium, cut it with enzymes, and then use other enzymes to paste them together, and you could put that into a bacterial cell. So you could take the human cell, the human gene, you could take the bacterial cell and the bacterial gene and recombine them together into the new bacterial cell. Now we have a bacterial cell that will make the human protein. So let's say that the gene that we took out of the human cell was a gene for insulin, and we put it in a bacterial cell. This bacterial cell will now make insulin. That DNA will be made into mRNA, which is then made into insulin. So this bacterial cell will become a little factory for insulin. Why on earth would we want a bacterium to start making human insulin? Do bacteria use insulin? Remember, insulin is for controlling the amount of sugar in the blood. Do bacteria have blood? No, they don't have a circulatory system. Insulin is useless to this bacterial cell. So why do we do this? Well, if you need a factory to make human insulin for people who cannot make insulin, that's pretty handy. And who can't make insulin? People with diabetes. So we have genetically engineered or genetically modified bacteria so that they start manufacturing this protein that they do not need. They build it up, we kill the bacteria, take their insulin, and give it to diabetics. It's great because the insulin matches human insulin and is very, very effective. In the old days, we would use insulin from cows or pigs and take that from their blood for diabetics. It would work, but it's not exactly the same as human insulin, and so it didn't do as good a job. This is exactly the right insulin because it is made from the human insulin gene and therefore has the exact right order of amino acids and is human insulin and will be highly, highly effective for the diabetic. So the uses, we have medicines like the insulin that I just described. Foods, we have taken genes from one species and put it into another species so that they can resist insects or fungi. So lots of resistance that we've engineered in plants. We've made food for animals that have extra nutrients in them. So lots of different uses for genetic engineering. Just to recap, the transfer of DNA from one species to another is called genetic engineering. The DNA made up from two separate species, when the enzymes are used to cut the DNA from one species and from another and to paste them together, the resulting combination of genes is called recombinant DNA. And finally, what is it that does the cutting? Enzymes.